Okay, chromosomes. How many chromosomes we got? Say again? 23 pairs? 46 chromosomes? We good with those? Everybody good with that? Where's the 23 from? Right, from the gametes? Okay. What houses the chromosomes? What cells house the chromosomes? Everything except for the red blood cells. Okay, so can we photograph the chromosomes? Are they that big? Yeah. Gene? What's the gene term? It's a portion of DNA that codes for a particular protein, and it's somewhere around 10,000. So the entire DNA strip itself doesn't actually do stuff, only the gene portions. And most of the DNA strip is pretty non-functioning as far as we're concerned. How do the bases of DNA and RNA differ? Okay, so DNA has a thymine, and RNA has a uracil, but the CG and A's are the same in both of those. So the adenosine, guanine, and cytosine. How do the sugars differ?
Second part of DNA has hydrogen and
Carbon 2 has no hydroxyl group on it. Missing an oxygen. What's phosphoester? Bless you. What else do we need with that? There's a phosphate. Carbon and then something else over here. So one of these phosphate, or the phosphate, is doubly bonded to one of these oxygens. And so it looks like an ester, except I've got a phosphate substituting for my one of my carbons. And why are they important to metabolism? Yeah. So adenosine diphosphate and adenosine triphosphate, the give and play between the two of those things are what constitutes metabolism for you. So losing a phosphate, gaining a phosphate. Draw and name one of the 4 nucleotides and determine what its pair is. So what do we got drawn? So we need a phosphate. And where's that going to connect on my deoxyribose? No? Yes? Was it bottom and then 3? And then just 1 down. That's right. So how come there's only 1 hydroxyl group on the sugar? On a really poorly drawn pentagon there. Why is there only 1 OH? The OH on this was used to make a water molecule, so the phosphate and the ribose could connect to each other. In the same situation over here, there used to be a water molecule between those, an OH on the ribose and an H on the nitrogen. So a water molecule disappeared. Then this one right here is missing because it's DNA. There isn't supposed to be an OH there. And so tell me what this is supposed to do. Attached to the next phosphate group that allows the chain to continue. So what carbon am I pointing to there in blue? Yeah. And so where's carbon number 3? Jeez. That one's carbon number 3. So when we're talking about the chain sequences of the bases, they connect from carbon number 5 and carbon number 3. So we'll end up having this, there'll be a link up here to a carbon number 3, and then this guy down here is going to link to a phosphate group which is connected to carbon number 5. And so this is cytosine, so this guy's going to connect to guanine. So the 5 prime, what's that referring to? Actually, it's not ribose. And when we're talking about DNA, we usually consider that to be the top of the leading edge of DNA. What are the single strands held together by? So the 2 DNA single strands are connected to each other, hooking up with little hydrogen bonds, but lots of little hydrogen bonds. And then what provides the structural support for the DNA? Phosphate sugar backbone. In the same way that the amino in the acid part in a protein is the backbone for the protein, the phosphate and sugar provide the backbone for the DNA.